Okay, so now we are moving on. We've talked about B cells. We talked about B cell in the process of being developed in hematopoiesis, and we talked about B cells producing antibodies. So now we're going to go back and put these kind of together and discuss more about how B cells actually do develop and how do they develop their B cell receptor that makes them specific for one antigen. And so B cell development is all going to occur in the bone marrow. And then, of course, B cell activation is going to occur in what we call the periphery, your body. So there are going to be stages of B cell development that are going to occur in the bone marrow. And then there's going to be stages of B cell activation that is going to occur in the periphery. Now, with respect to in the bone marrow, it starts, as we said before, with hematopoiesis. So you're always going to have a stem cell. Now, this, of course, is going to be that pluripotent stem cell, which then becomes the lymphoid progenitor cell. And this lymphoid progenitor cell is going to become a pro-B cell. Now, keep in mind, it could have become a T cell or an NK cell. Now, this pro-B cell is going to become a pre-B cell. And then it's going to become an immature B cell. And then it's going to become a fully mature in the periphery. And then this mature B cell can become a plasma cell after activation and differentiation, or it can become a memory cell after activation and differentiation. So this is sort of just the general overview. So in the periphery is where you get T cell activation and clonal selection. Now, some things we have to talk about before we talk about this whole process in general is specific molecules that are going to be important for B cell development. And so we have on the surface of B cells, a marker for B cells that's called CD19. And this B cell marker is going to be on the B cell through its life. So it starts when it first decides to become a lymphocyte. And then when your B cell decides it's not going to be a T cell, or an NK cell, it's going to be a B cell, it goes to the Bro-B stage, and that CD19 surface molecule stays with the B cell through its entire development process. Now, another molecule that's very important is RAG. Let's keep this. RAG is recombination activating gene, and RAG, remember, was very important when we were rearranging the B cell receptors, the heavy chains and the light chains, and we're doing the VDJ rearrangement. The deoxynucleotide transferase activity was also present during rearrangement, and so you can see that both of these are present during the pro stages and the pre stages, but as soon as you become an immature B cell, those genes get turned off because you've already made your heavy chain and light chain, and you don't ever reactivate RAG or TDT activity. Now, the other thing to keep in mind is that the heavy chain rearrangement occurs first. And so this is going to occur right after the pro-lymphocyte decides to become a B cell, and the heavy chain rearrangement begins. After heavy chain rearrangement, you are going to have the heavy chain on the surface for the remainder of the life of that particular B cell. Now, we also are going to have something called the pre-B cell receptor, and this is going to have a surrogate light chain. So we already made the heavy chain, and then the heavy chain is going to bind with that surrogate light chain and make the pre-B cell receptor. When this pre-B cell receptor is made, then you can get your light chain rearranging. And your light chain rearrangement is going to occur in the pre-B cell stage. So heavy chain in pro-B, light chain in pre-B cell. And then the light chain on the surface will be for the remainder of the life of the B cell. And the first immunoglobulin on the surface, of course, is going to be that IgM on the surface of the developing B cell. All right, so let's walk through these stages. Now I'm going to use a few acronyms, I guess. So CR is chromosome. And so we're going to start first again with our heavy chain. So we'll start over here with our pro-B, and we'll have our heavy chain rearrangement. And then we're going to have our pre-B, and this is where we'll have our surrogate light chain. So I'll call this heavy chain. And then we have our heavy chain plus surrogate light chain. And we also have in our pre-B our light chain rearrangement. And when I talk about rearrangement, this is that VDJ rearrangement, or VJ if you're a light chain. Now let's go in a little bit more detail. So our pro-B is going to be the heavy chain rearrangement, and it's going to be on chromosome 1. Chromosome 1, it decides, can be maternal, can be paternal, it doesn't matter. And if it's successful, you will move on. Oops. Now, if you get a failure, then you'll have to do a heavy chain rearrangement on chromosome 2. CR1 versus CR2. And CR2 would be the opposite. If you did maternal first, CR2 would be paternal. And then if it's a success again, you go up here. And if it's a failure, undergo apoptosis. So this B cell development cannot move forward unless you have a successful heavy chain rearrangement. And then you move to the pre-B cell stage. And the same thing's going to happen in the pre-B cell stage. So the light chain rearrangement is going to be on kappa on chromosome 1. Again, maternal, paternal, doesn't matter. And then if you're successful, you're going to have a successful T cell receptor. If it's a failure, it's going to go on the kappa chromosome 2. And again, if it's successful, it goes here. If it's a failure, it's going to go to the lambda light chain on chromosome 1. And again, successful, failure, lambda light chain on chromosome 2. And again, successful or failure. And failure again would be apoptosis. And apoptosis with respect to lymphocytes is called clonal deletion. Okay, so now you have a successful T cell receptor. And so it does have to go through another process, which we'll talk in more detail in just a moment, but it is referred to as negative selection. Now this is going to be central tolerance. And the purpose is to get rid of clones of B cells that bind to self-antigen in the bone marrow, because you don't want to release those B cells into the circulation. So if central tolerance or negative selection is a success, then you release your B cell, your mature B cell. If negative selection is a failure, your B cell is specific for an antigen in the bone marrow, then your B cell has the option to rearrange only the light chain. And so it will do a recheck. And if it's successful, it makes a successful T cell receptor, and it'll go through negative selection again. If it is a failure, it'll undergo apoptosis. So there are many checks and balances with respect to the rearrangement of your B cell receptor and whether or not it becomes a successful B cell. And then this is mature. Okay, so now let's just talk about central tolerance a little bit. So central tolerance in essence occurs in the primary lymphoid tissue. And the only two primary lymphoid tissues are the bone marrow, and this is for B cells. And it's going to be the thymus for T cells, which we'll talk about when we talk about T cell development. And so central tolerance is basically we have these cells maturing in these compartments. There are self-antigens, self-proteins around. Can these B cells recognize them? And if they can, we have to get rid of them. So within the bone marrow, there's going to be two different types of antigens. There's going to be cell-associated antigen or soluble antigen. So if you have a cell in the bone marrow that has on its surface these little, we'll make them a color, little green circles, these are cell-associated antigens. And if a B cell, an immature B cell, and its B cell receptors can bind to those self-antigens, then the B cell undergoes apoptosis. That means the B cell is specific for some cell-associated antigen. Now, if a B cell sees soluble antigen, so these will be purple circles, this time your immature B cell is binding into its B cell receptor, these soluble antigens, not cell-associated. And instead of undergoing apoptosis, this B cell becomes anergic. and it's going to increase Fas on its surface, and so it will be killed by Fas ligand. And so it can be killed by a T cell, it can be killed by a natural killer cell, anything that expresses Fas ligand, and that B cell will also eventually undergo apoptosis, but it's not an immediate apoptosis. Now, if you have a B cell, that does not recognize self-antigen, either soluble or cell-associated, it gets released. Oops. And it goes out into the periphery. Now, there also is peripheral tolerance. So there's central tolerance, which occurs in the bone marrow, and then there's going to be peripheral tolerance. So once you get released into the periphery as a B cell, the B cells have a half-life about 3 to 5 days. And what that means is that this B cell has to travel through your body, go from secondary lymphoid organ to secondary lymphoid organ to try to find its antigen. If it does not find its antigen within 3 to 5 days, it will undergo apoptosis via that Fas ligand type of interaction. And so how central tolerance occurs in the periphery is if you do release a potentially self-reactive B cell, the B cell will potentially become activated because your antigens are everywhere. And so you want to make sure that the B cell is not going to be able to proliferate and attack yourself. So you have to have what we call peripheral tolerance. And so we have, in our periphery, we have some checks and balances. And so I'm going to split this in two. We're going to have, in the case of an infection, and then we're also going to have in the case of no infection. So in the case of the infection, you have your dendritic cells. And your dendritic cell is going to have MHC class 2 on its surface. It's going to have some sort of virus antigen peptide. It's going to activate your CD4 T helper cell. That's going to recognize that antigen. And then this CD4 T helper cell is going to be able to provide help to a B cell. So this is occurring in the T cell zone. And then you have in the B cell zone, you also have a particular subtype of dendritic cells. And this one's called a follicular dendritic cells. Now, this is an important note. An FDC is not an antigen-presenting cell. The FDC cannot interact with CD4 T cells. The FDC is just called a follicular dendritic cell because it has dendrites like a dendritic cell, but it does not take in antigen, does not process and present it. Instead, the FDC will stick antigens on its surface so a B cell can come around and see if they're specific for that antigen. So, we'll have a B cell, and it will have its B cell receptor, and it's binding to that red antigen, for example. And then the T helper cell, because it's activated, will help the B cell. And because the B cell is seeing antigen, it's seeing inflammation, and it's getting help, this B cell is going to divide and is going to form memory eventually. It'll produce antibodies and do all that stuff that B cells naturally do. Now, your no infection side, you'll basically have your follicular dendritic cell, so here's your FDC. It will be expressing some sort of self-antigen. And in this case, there is no T cell activation. And so this B cell that recognizes the self-antigen is going to encounter the antigen, have no help, and so it's going to undergo apoptosis. And so it's important to recognize that B cells have several layers of control of their activation, and partly because tolerance of B cells is not as rigorous as T cells, as we'll find out. And so you have to have all these other layers to make sure that you don't get a self-reactive B cell being activated in the periphery. So now for B cell activation, again, we go back to we have our dendritic cell activating our T cells. We have our B cell seeing antigen by our follicular dendritic cells in the B cell zone. And then what's going to happen is the B cell is going to get activated. So I just want to describe a little bit about what happens with your B cells. So your B cells are undergo colonal expansion. And you're going to make many of these B cell clones, etc. And then the B cells are going to start releasing IgM. Now, two, you're going to have what's called the germinal center reaction. And in the germinal center reaction is where you're going to have your T cell help. You're going to have isotype switching. And you're going to have somatic hypermutation. So isotype switching, you're going to go from IgM to IgG, for example. The T cell help is necessary for this. So the T cells are telling your B cell to do isotype switching. The T cell is going to tell your B cell to undergo somatic hypermutation, and this equals a better fit. And so, for example, if we have our antigen here and it's this red square, make two of them, you have our B cells, and they have on their surface the B cell receptor. And let's say this B cell is going to recognize this corner of this antigen. And let's say the B cell looks like this. After somatic hypermutation, in which the B cell does small base pair changes in its variable region, you now will get a B cell with a B cell receptor on its surface. I'm drawing it too far away. That's a better fit. So the B cell receptor will be here, and it might look now like a point. So it fits that point better. So it's a better fit. So that's somatic hypermutation. Now, once you have these B cells can form memory or go to the bone marrow be a plasma cell. Okay, and then finally, let's talk a little bit about the different types of B cells. We have our conventional B cells, which is what we typically think about, making IgG, IgE, IgM, etc., IgA. And these are going to be found in our secondary lymphoid tissues. They undergo gene rearrangement. They do that affinity maturation, class switching. Affinity maturation is somatic hypermutation. They cannot secrete antibodies spontaneously, and they rarely make antibodies against self-antigens. We have T-independent, make that a different color, T-independent B cells. And these B cells are mainly in the spleen. They don't have very much gene rearrangement, and they don't have any ability to do affinity maturation, class switching, no memory, etc. We have our B1 natural antibodies, and these are in the not associated with the periphery too much. The peritoneum cavity is where they are. And again, they have limited gene rearrangement, and they can spontaneously secrete antibodies, and they tend to be those that are more commonly associated with self-reactive antibodies. And that is the end of this lecture. Thank you.